All right, so we're going to just hope that it stays. Oh, wait. Okay. So Progeria, you can barely see it, but Progeria is the reverse aging or the Benjamin Button syndrome. In this case, individuals who are impacted with this condition usually die in their early teens, and it's a result of just a mutation in one gene, a result of a mutation in just one gene. So this isn't even at the chromosomal level, but the whole purpose of genetic studies is so that we can see what the impact of each of these genes or chromosomes has. In this case, the one gene that causes this disease or this condition is the laminin A gene, which ultimately impacts the structure of your nuclear envelope. Where's your nuclear envelope? Outside the nucleus. So this structure is very weak, so guess what's not protected? The nucleus, the genetic material. Okay? The cells age much quicker, and as a result of this premature aging, they expire sooner. Why is this condition usually not propagated by these individuals? Why don't these people pass this condition on? They don't survive long enough to reproduce, so they are usually selected against. Yes. So if they die in their early teens, they haven't hit puberty. Body's not mature enough to produce the gametes necessary for reproduction, so this condition does not get passed down. So this is the feature story in your text. So a couple of things I want to remind you of. First of all, what a gene is. By definition, and we've said this over and over this semester, a gene is just a segment of DNA. And usually when we reference genes, we talk about them as being like a line. So each of these lines here represents a gene. A chromosome, on the other hand, is a large segment of genetic material or DNA, and it has multiple genes. Okay, so each chromosome can have many, many genes, anywhere from a few hundred to a few thousand genes. So a gene is just a short segment of DNA, and each gene codes for one. What does a gene code for? Protein. Each gene codes for one protein, and when you have a chromosome that has multiple genes, all of those genes each individually code for a protein. Yeah. Yeah, a gene is the instructions for protein. We use genes for protein synthesis, which is what? Creating proteins, yes. So my chromosome that has multiple genes has the ability for multiple proteins versus just one gene is used to make one protein. Okay? So genes versus a chromosome as in terminology sense. Now, the topic that this takes me into is what most of this chapter is going to focus on, the difference between linked and unlinked genes. Linked and unlinked genes. Linked genes are closer together on a chromosome. Unlinked genes are not close together on a chromosome, or may even be on separate chromosomes. So they're considered linked if they're very close together. And I mentioned this last lecture when we talked about red hair and freckles. Many people believe that those genes are linked and that they have to stay together. But do they have to? No. What process could separate them? During meiosis, what happens? Crossing over. So if these two genes are really close together and what we considered linked, what we're basically saying is because of their location, even if crossing over does occur, they're probably going to stay together. However, that does not mean that they have to. It just means that there's a higher chance that they will versus they won't. So the closer genes are on a chromosome to one another, the more likely they are to be linked. Okay? So one more time, that crossing over, that recombination occurs during meiosis, specifically during prophase 1. What am I forming during meiosis? Sex cells, or my gametes, if I'm going through meiosis, and I'm going to come back to that in a minute. So we use chromosome maps to calculate the probability that genes are going to stay together or separate, for that matter. So we have what we call a chromosome map. On a chromosome map, you have a chromosome, and it maps out all the individual genes on that chromosome. The distance between genes is measured and recorded in what we call map units, or MU. Each map unit is equivalent to a 1% chance that those genes will recombine or will separate. So for example, let me interpret what I just said. Looking at gene A and B, they are separated by a distance of what we record as 6 map units, which means, if interpreted correctly, each map unit equals a 1% chance of recombination. So there is a 6% chance that these two are going to do what? Recombine or separate. But there's a 94% chance that what? They won't. There's a 94% chance that they won't. 6% says that they'll separate. 94% that they won't. If I'm looking at genes D and E, they are separated by a distance of 52 map units. So how could I interpret that, or how would I? Yeah, there's roughly a 50% chance that they're either going to stay together or recombine. Question, do they have to recombine? No. Does crossing over have to occur here? No. This whole process is what we consider random. Random. So if you had this set up here, this chromosome map, and it said which two genes would most likely recombine, A and B or A and D, you would answer, why A and D and not A and B? They're further apart, so the distance is measured by map units. Those map units are the equivalent to the percentage that they are going to recombine. A and B are very close. A and D are further apart. What cells does this crossing over occur in? Your sex cells, your gametes. I just asked you that question. Your sex cells, your gametes. Does this happen in your bone cells? No. No, it doesn't. Okay? Bless you. Or cough you. Last time. Unlinked versus linked. Genes are considered to be unlinked if they have a large distance between the two. So the chances that they will separate as a result of recombination are pretty high. Linked genes are very close together on that chromosome, and so the chances that they separate during recombination are decreased or smaller. They tend to stay together. Now, what cells in our body does recombination occur in? Sex cells. Sex cells or gametes. And so what's the process of recombination? Crossing over. When does that occur? What phase of meiosis? Prophase 1. Fantastic. Now, this is happening in you right now as you're creating gametes. How does this crossing over affect you? How does it affect you? You're creating all these gametes with all these different combinations. How does this impact you? Are you changing right now? We are? So the genetic material in my cells is changing as a result of my eggs recombining. Does this impact me? No. It impacts my potential offspring. And why do I say potential? I may be making a ton of gametes, as are you, that will never become kids. This recombination, this crossing over, all of this right here that's happening inside of you doesn't impact you or doesn't change you. You're already set. You're already formed. Make sure you know what we're referencing when we talk about these processes, okay? Yes, this occurs in you constantly once you hit puberty, but it doesn't impact you if there's changes, if crossing over occurs or doesn't occur. It only matters if that gamete is fertilized and becomes an offspring. So let's talk about the term sex-linked. And what does it sound like? What does it make you think? Sex-linked. Sex-linked genes. Okay, it has something to do with gender. Fantastic. I taught you 4 terms a few lectures ago, and I'm going to remind you of these 4 terms because you need to know the difference. The first 2 terms were somatic versus sex cells. What's the difference between somatic and sex cells? Somatic are your body cells, and your sex cells are your gametes, your sperm or eggs. What process do your somatic cells undergo for cell division? Mitosis. What about your gametes? Meiosis. Okay? So if I said bone cell, you would say somatic mitosis. If I said brain cell, you would say somatic mitosis. If I said sperm cell, you would say gametes, meiosis. Do you see what I'm doing right there? If you know what type of cell you're dealing with, you can answer this question. Now, first thing I asked you to do at that point was distinguish between the 2 types of cells in you. Second thing was the chromosomes. How many sets of chromosomes do you have? 23 pair, right? You have 46 or 23 pair, yes. Now, I told you that we have different names for these chromosomes. Chromosomes 1 through 22 are considered autosomes. Chromosomes 1 through 22 are considered autosomes. But chromosome set 23, those are your sex chromosomes. Those are your sex chromosomes. So chromosomes 1 through 22 are your autosomes, and chromosome set 23 are your sex chromosomes. So I taught you those 4 terms: somatic cells, sex cells, autosomes, sex chromosomes. And then I asked you this. What cells in my body have chromosomes 1 through 22? All your cells do. But what cells in your body have chromosomes set 23? All of them. All of your genetic material is in all of your cells. It is. When you create gametes, though, you just get 1 set. But it still has chromosomes 1 through 23. And I made sure when I finished that aspect of that lecture that you understood the difference between those terms. You have to know that for these tests. So when we look at this slide and it says sex-linked genes, can you tell me probably which chromosomes I'm dealing with? Which chromosomes I'm dealing with? Pair 23. The question doesn't say sex cells. It says sex-linked genes. The only place I'm going to find sex-linked genes are going to be on my sex chromosomes. They're going to be in my sex cells, They're going to be in all my cells. But the genes are going to be on my sex chromosomes, which are on pair 23. And we know all of this because of this organism here. This is a Drosophila species, the fruit fly. And this fella here has the same, and lady, has the same sex chromosomes you and I do. So why do we test on them and not us? They're model organisms. Fantastic. So when we do sex-linked studies, we use fruit flies because they reproduce very fast. Nobody cares if they die. They're easy to maintain. Okay? So they're our model organism. Chickens don't have XYs like you and I do. They have ZWs. So not every species has XY like us or has similar genes on those chromosomes like us, which is why we chose this organism, which is the fourth aspect of why it's a model organism. So for females of these species and our species have two X chromosomes, and males have both X and Y. All right? Going on this, and I'm still on sex-linked, but we already said sex has something to do with gender, which we do have each of those chromosomes in our cells. But what I have here is the probability that your offspring will be a certain gender. And what is the probability? Why is it 50-50? Okay, so here's mom's eggs. Her gametes separate. Remember, during meiosis, they'll line up, and then they will separate, one going into each gamete, or segregate. So as her gametes form, she had two copies. As her gametes form, there's one copy in each one. And dad will do the same. His X and Y will be together, and then when he goes to form gametes, they will separate. So mom will have just X's, and dad will have both X and Y. So we set up this Punnett square here with mom's gametes and dad's gametes. And as you cross them, you can see that there's a 50% chance that you'll conceive a boy or a girl. We did talk about increasing the likelihood in a separate lecture, and those are all environmental factors. But the hardcore probability is you have a 50-50 chance whenever we look at that. Now, the sex chromosomes. So again, which pair am I dealing with right here? 23. You have an X chromosome, and some of you have a Y. The X chromosome and the Y chromosome are not the same. You can visually see right here the difference between the two. The X chromosome is very large. The Y chromosome is very small. Which one do you think houses more genes? The X chromosome. The X chromosome has roughly 2,300 genes. And guess what? Raise your hand if you have an X chromosome. All of us do. Because all of us do, these genes aren't specifically sexually geared towards you being a male or a female. Because if that were the case, all of us would be girls. So the X chromosome has roughly 2,300 genes, and they cater to non-sexual aspects because we all have X chromosomes. The Y chromosome, though, is much shorter, and it only has a few genes. A few. But most importantly, it has one gene called the SRY gene, and that SRY gene is the gene that is responsible for boys developing like boys. So, no matter what, if you have a Y chromosome, what are you? A boy. If you have a Y chromosome, you are a boy, due to the SRY gene that is housed there. The SRY gene is the gene that's responsible for males developing into males. If you don't have a Y chromosome, you are a female. Okay? So again, this is just pair 23. You can see the marked difference in size. The X chromosome is very, very large and houses thousands of genes, versus the Y chromosome, which is very small and only has a few, but most importantly, it has the SRY gene, which tells a boy to become a boy. Now, we've talked about what a sex chromosome is. We've talked about how to determine the sex of an offspring. We've talked about what material the sex chromosome has or houses. Now let's talk about what I mean by sex linkage. Sex linkage is where a trait is specifically tied to one of your sex chromosomes. And generally speaking, I'm talking about the X, because the X is the one that's going to have the most genes. So when we say sex-linked, I'm talking about one of those 2,300 genes on the X chromosome and its alleles that are associated with it. You need to be familiar with two conditions: colorblindness and hemophilia. These are two sex-linked conditions, and I'm going to walk us through examples with both. Okay? So what we have here, the first one that I'm going to walk you through is colorblindness. I'm going to let capital C represent someone who can see normal. I'm going to let lowercase c be someone who is colorblind. When we look at these characteristics, these sex-linked characters, you'll find that most sex-linked conditions manifest themselves in boys as opposed to girls, and I will show you why. Oh, why? But that wasn't supposed to be a pun. But it's because of the Y chromosome. So let's say that I have a mom, and she herself can see normal, but she carries a colorblind allele. Remember, mom has two X's, and the gene for colorblindness is on the X chromosome. So there are different choices. You can either be colorblind or not. Those are the alleles, and she carries one of each. Dad, on the other hand, he sees normal, but that same gene is not on his Y chromosome. So he just sees normal. How do I know this is dad? It has a Y. If there's a Y, it's a boy. So when I do my Punnett square, I'll put mom here. So mom's gamete, her other gamete, her recessive one, because she's a carrier. And then I'll put dad across the top. He's dominant, but he has a Y. When you do a sex-linked cross, we always represent the sex of the parent. So you'll always have X's and Y's. You will not just put, I have colorblind, because then you would have nothing here for the Y. Okay, so we'll always use X's when it's sex-linked. You need to know that because you'll have one on your quiz today. But of course, I'm going to walk you through how to do this correctly. Now, here is my first potential gamete offspring combination. So you cross these just the same way. And you interpret them the same way. So this genotype here, so in my genotype analysis, how many do I have that are homozygote dominant and can see color? 1. How many males do I have that are dominant? 1. How many females do I have that are carriers? 1. How many males do I have that are colorblind? 1. So my ratio would be 1:1:1:1 for genotype. It's the exact same way that we worked them out last week. Now, is this kid colorblind or normal? Normal. Male or female? Female. So my first phenotype is a normal female. How many do I have? One. One normal female. What about this kid? A normal male. How many do I have? One. What about this kid? It's a carrier female. And there's one. Why can a carrier not be a male? He only has one X chromosome. So what is this kid? It's a colorblind male. So how would I write my phenotypic ratio? Right. It would be 1:1:1:1:1. Okay? And again, why am I using the X's and the Y's in this problem? Because it's sex-linked. Because it's sex-linked. Okay. Now, that was colorblindness. Here is a Punnett square for hemophilia. So, hemophilia, it tells you here that capital H designates a normal allele, and a lowercase h represents the hemophilia allele. Hemophilia is the inability to clot blood. So you have clotting factors that you naturally produce, and if you are an individual that has hemophilia, your blood does not clot. So if you cut yourself, normal people, when you cut yourself like a paper cut, you can put pressure on it and it clots and forms a scab. But people with hemophilia, they don't have the ability to clot. So it just continues to do what? Bleed. Okay? And that's a true condition. We can treat it now if we know you have it, but in the past, many people have died as a result of this because they bleed over what normal people who have the normal clotting ability would just heal from. But they bleed and continuously bleed. So, I know that this is sex-linked, and I can tell that because I see the X and Y's. So, even if I didn't know that, because there's X and Y's there, I know I have to be dealing with sex chromosomes. Okay? Now, it tells me here that I have this parent, and it says X and then X with the lowercase h. Is this my mom or my dad? How do you know? 2 X's. But is she normal? One of those X's doesn't have a letter. So what should I be assuming? She's a carrier. So is she normal? She herself does not have hemophilia. So what letter am I assuming is missing there? A capital or a lowercase? Capital. Fantastic. If we can analyze these together, when you do come across them yourselves, then you're like, okay, I can remember that. So in this case right here, I have a mom who is not a hemophiliac. However, she carries the allele for it. And I have a dad who is a hemophiliac. But why doesn't the Y have a letter? It's only on the X chromosome. Okay? So here I have dad, his potential gametes, here's mom's. So this kid right here, male or female? Female. Affected or carrier? Carrier. So I have a female carrier, 25%. And you can see that this is represented here as well. So my genotypic and phenotypic ratio. What do I have here? A female who has hemophilia. So 25% chance for that genotype, and 25% chance that I have a female with hemophilia. What do I have here? a normal male. Normal male, 25%. Normal male, 25%. And what do I have here? A male with hemophilia, again, 25%. So it's analyzing your cross once you've done that cross. And I'm using X and Y's to indicate that this is a sex-linked characteristic. Okay? Now, of course, these Punnett squares are fun, but a lot of times we may not have, we may be talking about things that have happened in the past. And in this case, we would use a pedigree chart. A lot of times when people hear the term pedigree, they think of dog, which I get it. But like your 23andMe and all of those chromosomal and ancestry type things that you do now, those are being used to create pedigree charts. That's what they're doing. Okay? And by you paying, you're giving them your genetic material so that they can help put everything together and create one big family tree. All right? However, they do give you some information about yourself too, so that's always good. In this case, we have a pedigree chart for hemophilia. So again, this is an X-linked characteristic. We just spoke about it on the previous slide. But the reason this pedigree chart was constructed initially is because there was an issue with hemophilia. People not being able to clot their blood, and they were trying to figure out where does it originate. Now, when it originated initially, and this individual had their offspring that had hemophilia, and that baby, well, it was a grown man at that point, but bled, and they couldn't figure out why it wouldn't stop, they didn't know about hemophilia. They just said he had a bleeding disease. But as we started to check through generations, we saw that after that, it showed up more and more prevalent. Looking at this pedigree chart, what time frame would you estimate we're measuring here? What time frame? How many? Okay, great time. I get that. Is how many years are being represented here? I have 6 generations. Okay, so if we said 600 years, that would mean that each generation was alive 100 years before reproducing. So if this started way back in the day, what was an average age to have a child? Earlier or later in life? Earlier. So let's say like 20s. So if each of these generations is divided by around 20 years, this roughly gives me about 120, on the far side, 150 years of data. Okay? Now, it started here, and you can only provide information on individuals that you're knowledgeable of. So when I look at this family tree, it starts at a certain place that might seem a little bit confusing. For all pedigree charts, there should be a key or a legend, but generally speaking, every time there's a circle that represents a female, a square represents a male. If that circle or square is partially shaded, that individual is a carrier. And if that circle or square is fully shaded, that represents someone who is afflicted with that condition. So when I look at the beginning of this family tree or pedigree chart, it starts with generation 1 and 3 males. And so a lot of people are like, what? Even my last class said, wait, it starts with 3 guys. We only know about the guys. We don't know. Obviously, if they reproduced, there had to be, but we don't have data on her. So we don't just throw some girl up there and say, oh, she's the mom. We don't know. We don't know who she was. And we don't have any data on her. But we do know that because these gentlemen were part of royalty, we have access to them. So here they are. And each of these lines here represents their offspring. So this gentleman here had 1 boy and 1 girl. This gentleman here had 1 son, and this son reproduced with this gentleman's daughter. Do you see how I'm reading that? this gentleman here had 2 sons, and his sons reproduced later on with the 3rd generation down the line. So these 2, this son and this daughter that reproduced, they had Queen Victoria of England. And of course, at the time, she was perfectly normal. She had no idea, neither did her parents, that she was a carrier for hemophilia. Well, Queen Victoria reproduces, and here are her offspring. So you can see here they reproduce. She has 1 daughter who is a carrier. This right here, this kink, tells you that this is not her kid. This is the gentleman who reproduced with her daughter. So this kink says that that's not her kid. Okay, so this is her spouse. She had 1 son, and why does it have that like ... He didn't reproduce. Okay, or that we have knowledge of. She had another daughter who went on to have 3 boys and 2 girls. She has a daughter here. What do you think? Probably expired. That's the way that I would interpret that as well. She probably didn't survive very long or long enough to reproduce. Then we have a son here with just a short, we don't know. We don't know if he didn't reproduce. We don't have the data on that. Why don't I just put some stuff up there? We don't know. She has a son who has hemophilia. This is the bleeding disease son. Then she has another daughter who's a carrier who reproduces with the brother of that other kid. Okay? Anyways, here we have our 2 carriers, and we have 1 who's afflicted with hemophilia, and it's a male. What happens in each generation after? What do we see about the incident, the number? We see the number increasing. Once that allele was introduced, and we don't know if these parents were carriers or if this was just a random mutation. But after it started, each generation after, it became more and more prevalent. It became more and more prevalent. You do pedigree charts so that you can kind of calculate or see throughout the history how often this disease is shown up. Okay, so pedigree charts. You'll need to be familiar with how to interpret one. You will not have to create one, but just how to read through it and understand what it means. For X inactivation, how many X's do we need? Why do we know we only need 1 X? Raise your hand in here if you have an X. We all do. How do we know we only need 1? Okay, there is only a 50-50 chance that you can get 2. But how do we know you only need 1? Because males only have 1. And if you needed more than 1, then they wouldn't be here. Okay? Now, as a female, how many do you have? You have 2. Okay? So, you have your X chromosome, and do you remember roughly how many genes are on your X chromosome? Roughly 2,300. So I have an X with 2,300 genes, and of course, because I'm a female, I have 2 copies of that X. And how many genes are on that second copy? 2,300. What's the problem here? I have too many genes. I have too many genes. Every gene that was on the first X is also on the second X. And what do genes give me the information for? Making proteins. So if I double the number of genes, what am I doubling? Number of proteins. And too much of a good thing is bad. So in females, during embryonic development, 1 of their X's is inactivated. Randomly. Because they have 2 X's. 1 of them is randomly inactivated. When is embryonic development? When you're a super tiny, tiny, tiny baby. This was you. This was you when you were a zygote. This was when sperm and egg met. You probably don't remember. It's not a big deal. I'm not asking for family photos or anything like that. I'm just saying that at one point you were one cell. And if you were a female, you would have 2 X's at this point. On this diagram here, each of these X's is a different color. One is orange and one is black. And that is significant because the next slide is going to give you the example of a calico cat. The calico cat, their color is sex-linked. So the color of that cat is determined by their X chromosome. How do we know that doesn't happen for humans? Because we all have similar color patterns. Okay? Let me stand by. Because if our color was X-linked, we would look like calico cats. Females would. Males would not. Hear me out. So here you were, just a tiny little zygote, 2 X's, and then this zygote started to go through mitosis. And when you were this size, roughly 16 to 20 cells, so at this point we call you a blastula. No, morula, then blastula. Anyways, you're super small. Like you don't even have eyes yet. One of these X's in each of the cells is randomly inactivated. And so you can see in this cell here that the black X is left on, but the orange one is inactive. And here the orange one is left on, but the black one is inactivated. And it's just random which one is turned on and turned off. But the way we inactivate it is we tightly coil it. We bring it up really tight so no enzymes can access it to make copies with it. So when we really tightly wind it up, we reference it as a bar body. A bar body is what you call a really tightly wound X chromosome. A really tightly wound X chromosome. So here I have one of my X's. The black one is on, the orange one is off. So from this point forward, every single time this cell here goes through mitosis, what color will it represent? Black. And from this point on, every time that this cell goes through mitosis, what color will it represent? Orange. So if the cells in that area right here represented black, guess what color this would be? And if the cells in this area represented orange, guess what color would be there? Orange, which would lead us to having a variegated pattern. Because can we control where these cells are going to go? No, but wherever they go, wherever this cell is, I can tell you it will be orange. And wherever this cell is, I can tell you it will be black. And all of its descendants will all be black. Why don't we do this in males? They only have 1 X. That's all you need. But females have 2. So we need to inactivate 1. And that's done randomly. So here, I can tell you that the black is the bar body and the orange one is the one that is expressed. And the reason we know that is because the color of this cat is sex-linked. Now, why are there no males that have that pattern? They're either going to be orange or black because they only have 1 X. So there are no male calicode patterns. They are a solid color. This format of color distribution is what we call variegation. In humans, once again, if as a female, our skin color was determined by a sex chromosome, we would look like that. Just whatever 2 colors we had. So we would have this like crazy pattern, and it would be cool because it would be unique to each of us, but still. So that's as a result of X inactivation because you only need 1 X chromosome. Now, this section right here is going to sound really, really similar to what we spoke about last week when we talked about deletions and insertions. But last week when we talked about them, we were talking about at the gene level. So we were talking about the deletion or insertion of one nucleotide, which would change one or a few codons that would result in the change of an amino acid. But this week, each of these letters doesn't represent one amino acid. Each of these letters represents an entire gene. Each of these letters represents an entire gene. And what is a gene responsible for doing? Making proteins, having the information to make proteins. So if I don't have a gene, if that gene is deleted, as seen in this first example, tell me what occurs. If gene F is deleted, what impact does that have on me? If gene F is deleted, what impact does that have on me? The chromosome does change. What impact does it have that I've deleted a gene? I don't make that protein. I don't make protein F. If I take away gene F, I don't have protein F. That's a big deal. That's a big deal. But I do want to remind you, last week we talked about the fact that some genes are what we call coding genes, which means they code for a protein, and some of them are called non-coding genes, which means that they don't code for a protein. So what is the impact of this? If gene F is a coding gene, I'm not going to make that protein. And if that protein is necessary for life, guess what? I'm dead. But if it's something that my body can handle, then I'll survive. So the true answer is, well, it really just depends. The fact that this is being deleted, all of it. Her question was, is that random too? The answer to that is yes. You have no control over whether that's going to occur. Now, if that chromosome mutates inside of you right now, and it just happens in one cell, is that really a big deal? Not really. But what if this happened when you were a zygote, just one cell yourself? Then it's a big deal. Then it's a big deal. Okay? Another example of a chromosomal abnormality is duplication. And duplication is the opposite of deletion. In this case, I'm adding, I'm doubling. So here I have my normal chromosome with genes A through H, but D and E are duplicated. You, at a homeostatic level, have the exact amount of genes to make the exact amount of proteins you need to survive. So what have I done here by doubling these genes? I'm doubling the amount of proteins there. Good or bad? Bad. And it really depends on what protein that it makes. Okay? The next example, translocation, you and I technically touched on last week because it was on the examples of the mutations, and I said, I'm going to come back to that one because that's a chromosomal one. And we talked about the flippers and all of that. Okay? But what I have in this instance are two non-homologous chromosomes. What does that mean? They're non-homologous. They're not the same. Remember, homologous means I have the same chromosome but from a different source. So like chromosome 1 from mom, chromosome 1 from dad. Okay, non-homologous says I have chromosome 1 from mom but maybe chromosome 3 from dad. And the problem is, first of all, they don't even belong together. But not only are they together and they shouldn't be together, but they actually recombine. What do I mean by recombine? They cross over. What process did they cross over in? Prophase, that's the phase. What process? Meiosis. And during prophase. You are fantastically correct. Prophase 1. Yes. So they've crossed over. And here's my question. First question, actually. Did I lose any genes? No. I still have all the same letters there, so all the same genes are there. Second question, does order matter? Yes. How do I know? Andrea and I both have, first of all, Andrea and I both have only 4 letters that make up our genetic code, A, T, C, and G. We all do. But how do I know, just based on her and I, that order matters? We don't look the same. We're 2 totally different people. But what if our letters were in the same order? We would be the same person. So order does matter. So these genes are still here. So technically, if you analyze it that way, they could still make proteins. But does the order matter? Yes. And here we have a big problem. The example I showed last week, this baby didn't develop a brainstem. A baby without a brainstem is dead. That's a big deal. And then the last example is inversion. Inversion shows A through H together, but the E, F, and G switch orders. The genes are still there. They're still making protein, but does the order matter? Yes. So this is a big deal as well. Another issue, and you and I have talked about this today and in previous lectures, but nondisjunction. This is a chromosomal abnormality that could occur during the formation of gametes. What process am I talking about? Meiosis. Again, it's a chromosomal abnormality that could take place during the formation of gametes. It's meiosis. Remember, during metaphase, they line up in the middle, and then during anaphase, what should they do? Separate, so that each gamete gets the exact same amount of genetic material. But during nondisjunction, which can occur during metaphase/anaphase, instead of separating, what do? They stick together. And the problem with that is that because they stuck together, this gamete has too much and this gamete has too little. This gamete has too much, this gamete has too little. So you can see here, they didn't divide that they were supposed to, but then it got back and synced them. And now I have 2 gametes that don't have enough genetic material, and I have 2 gametes that have too much. Now, if you make these gametes, what impact will it have on you? None. None impact on you. You have already been created. What impact does it have if you don't have kids? None. The only time this becomes an issue is if these gametes are fertilized to create an offspring. This may have occurred in you thousands of times, but it didn't matter because you didn't have a kid. And that gamete wasn't fertilized to become an offspring. So you never thought twice about it. You didn't even know it occurred. This process of nondisjunction can occur during meiosis 1, between metaphase and anaphase. It could also occur during meiosis 2, because remember, during meiosis, it divides twice. In this case, it went through meiosis 1 perfectly normal, and it proceeded. These 2 went perfect, and we have 2 nice, healthy gametes with the correct number of chromosomes. But during metaphase and anaphase of meiosis 2 on this side, 2 chromosomes stuck together that were supposed to separate. So now I have 1 gamete with too few and 1 gamete with too many. Again, if they are not used to fertilize, this has no impact. The changes in chromosome number, this is a slide of terminology. Do you remember what the prefix U means? Good or true? Fantastic. So when I say that this organism is euploidic or euploidy, that means that they have the correct number of chromosomes. So I put in parentheses 46 because 46 is the correct number of chromosomes for humans. So I'm trying to relate to something that you know. Do you recall what the prefix A was for? Not or lacking. So aneuploidy means what? It's not the right number of chromosomes. You either have too many or too few. So I put it up there as plus or minus 46. You're aneuploidic. Now, based on many discussions that you and I have had, if you're aneuploidic and you're a human, what probably is going to take place during embryonic development? If you have extra or not enough chromosomes, what's probably going to happen? It's going to be a terminal pregnancy, meaning that you're going to miscarry. More than likely. However, if that child does survive, and we're going to talk about that, if that child does make it, there will be complications. Yes. The third term is polyploid. Now, poly you already know means many, and then ploidy is referencing this set of chromosomes. So polyploid, you and I are diploid, which means we have two sets of chromosomes. Haploid would be just one, but triploid would be 3, and tetraploid would be 4. When you have that many chromosomes and you're a human, you're not alive. That's lethal in humans. Polyploidy is lethal in humans. But because this is biology and we can't just always focus on humans, although that is a bunch more fun, polyploidy is pretty popular in our plant population. Polyploidic plants have a greater success rate. They tend to be hardier overall, withstanding more intolerant conditions long enough to reproduce, so they're passing that on to their offspring. This may also be a part of a genetic modification, so something that was done in research. But having that many sets of chromosomes definitely leads to genetic variation. And genetic variation either makes you more fit, where you're selected for, or less fit, where you are selected against. So polyploidy is not a thing for humans. But here's aneuploidy, and as I mentioned, we were going to talk about it. And I started off at the very beginning with abnormalities in chromosome numbers for humans usually prevent embryonic development. And I put in parentheses, that means it's a miscarriage. Now, I also just said humans. This doesn't just apply to humans. It's to animals. Okay? If you don't have the right number of chromosomes, it's inconsistent with what your body or those cells can function with. The majority of aneuploidic offspring will be miscarried. However, Down syndrome is a perfect example of a situation in which there is an extra chromosome and this child still survives. So here I have a karyotype, which again is the organization of all chromosomes in order from largest to smallest, so 1 through 22. What do I call chromosomes 1 through 22? Autosomes. And an individual who has Down syndrome on chromosome 21, instead of just having 2 copies, they have 3. Hence the name trisomy 21. Down syndrome means that they have 3 copies of chromosome 21, so we reference it as trisomy 21. Now, look at the size of chromosome 21. Do you think that there are a lot of genes or a few genes? A few, which may be part of the reason why this child survives. Because there weren't a ton of genes there, and the genes that are there, we could work with having extras of whatever they were. Okay? Now, where are the sex chromosomes here? 23. So can you tell me if this right here is a girl or a boy? How do you know it's a girl? The 23 or the 23rd pair, chromosome 23, these two are the same. The only reason they would be the same is if they were both X's. If it were a boy, it would be 2 different chromosomes. You would have a long one and then a very small, short one. The reason most students miss this question is because they look at this and then they look at the picture underneath and see that it's a boy with Down syndrome, and they think the two are related. There's small things that you pay attention to and you can totally get. The purpose of the second picture is straight out of your book, is to show the incidence in which children with Down syndrome tend to be born after a mother reaches a certain age. So nondisjunction, the probability of nondisjunction increases in females around the age of 35, and it continues to do so exponentially each year after. If you become pregnant now and you're 35, you will be classified as what we call a high-risk pregnancy for the first 1 to 2 trimesters until we can make sure that your child is normal. The likelihood is that your child is normal. But because you're 35, there's a chance that you've mutated or that non-disjunction has occurred in this gamete, and there could be a problem. So what we need to do is we just need to monitor. Okay? So 35 is what we consider high risk now. Just so you know, 10 years ago it was 30. What we're tending to see is that the women who are waiting till later in life to reproduce have actually taken really good care of themselves. They feed themselves well, they've nourished their body. If you reproduce earlier in life, the likelihood is that your cells separate the way that they're supposed to. Later, it's just that the process is a little bit slower. But if you have good health and take care of yourself, you're really decreasing the chances that it will occur. My thoughts are that the people who wait till longer in life have either a really solid career or they've been trying to set up, so they've taken care of business. Does that make sense? And in the event that this is not that person, then I'm not speaking to population as a whole, but you'll tend to see that. And I would put money that within the next 10 years, this high-risk incidence rises to about 40. Because we're starting to see women have children later and later in life as they built their career and put their lives together and so on. Yes, ma'am?
There is a chance of, I know you say it's a high risk at 35, before that it's also like a possibility?
It's definitely a possibility. The likelihood is, though, that it will not occur. So have you ever heard someone say, like, your biological clock is ticking? That's truly what they're talking about. Like, as far as your metabolic rate and the way that your body operates, with age, it slows. Your metabolism slows. The efficiency of your processes slow. But we also know, with the science and technology we have now, how to better serve ourselves as women, as females, people who are potentially going to have kids at a later age. So you take care of yourself, the likelihood is that there won't be a problem. So before 35, you're not considered high risk. You could still have a child with an issue, for sure, but the probability is much lower. This slide here talks about the aneuploidy of sex chromosomes. Can you specifically tell me what I'm referencing, please? Aneuploidy of sex chromosomes. The wrong number for which ones? So what chromosomes am I dealing with? 23, pair 23. So what this is I have the wrong number of chromosomes for pair 23. The wrong number of chromosomes for pair 23. I have a female who, when she went to go make her gametes, instead of the X's separating, one gamete got 2 X's and one got no X's. So when this egg here with no X chromosome is fertilized potentially by a Y sperm, do you remember how many X's you need? You need 1, and there's none here, so guess what's going to happen to this kid? Expire. Here we have that same non-X egg, but fertilized by a female sperm. How many X's do I need? 1, and I have it. This young lady will survive with just 1 X chromosome. She will have complications. She has the wrong number of chromosomes. A female with just 1 X is considered a female with Turner syndrome. Females with Turner syndrome have a shorter stature. They're shorter. They're stockier. They have a webbed neck or a caped neck, which means that their chin and their shoulder kind of connect, so it's like a webbed look. You can search pictures, of course, on YouTube, or not YouTube, Google Images. Fingers are webbed, toes are webbed. She's just a shorter female. She herself is sterile, so she will not have produced gametes and successfully reproduce. Her mental capacity will vary on range, so she may have some severe neurological issues, or she may not. And sexually, she'll look like a female, but she will be very underdeveloped. Very underdeveloped. If this second gamete with 2 X's is fertilized by a Y sperm, so I'll have 2 X's and a Y. Do I have a boy or a girl? Boy, because I have a Y. One of those X's will be what? Inactivated. Fantastic. So at 2 X's and a Y genetically is what we call Klinefelter syndrome. Klinefelter syndrome is a situation where you have a boy that looks like a boy. He hits puberty later, so he has less body hair. Okay? Mental capacity is pretty normal, although it could range. Sterile, so he himself will not reproduce. And his testes tend to be underdeveloped, which leads to him being sterile. So he is a boy. This, I have a lot of people say, so does this mean that he's going to be gay? No. This does not mean that. Well, is this a hermaphrodite? No. A hermaphrodite, I think society has a huge misconception as to what a hermaphrodite is. But this is not an individual who is a hermaphrodite. A hermaphrodite, by definition, has 2 sets of gonads, testes and ovaries. Most hermaphrodites have no idea that they're hermaphrodites. They go their entire life either female dominant or male dominant, looking like a girl or looking like a boy. And they don't know that they're hermaphrodites until when they try to have kids. So they've had sex this entire time, but now they're trying to have kids. They don't produce gametes. They are sterile. So they're trying to have kids and they can't get pregnant. It's because if you're female dominant, your ovaries had a small influence of testosterone from the very, very small testes that are still inside your abdominal cavity, so you couldn't create or release eggs. And vice versa for males. Your testes, because you're male dominant, you have them, have been slightly influenced by low levels of estrogen, which have halted the production of your sperm, so you don't make gametes. That's what a hermaphrodite is. Most people don't know they're hermaphrodites. When people are like, oh, I thought a hermaphrodite had a penis and a vagina. No. That's plastic surgery, okay? Now, I'm not saying that there isn't some natural occurrences where that occurs, but it's highly unlikely. I've had many students throughout the years say, well, if I did have my own penis and vagina, I would have sex with myself. That's an excellent plan. You obviously don't understand the mechanics of sex, because in order to do so, you would have to obtain an erection. And if your penis was erect and your vagina was behind your penis, you would have to bend it. And it's just not going to work out for you. But, the thought was there. I appreciate the idea and your ability to take a risk in our class. So I've dealt with questions like these all along, but there's a difference. And the penis and the clitoris are the exact same organ. So you have one, and it's either going to be a penis influenced by testosterone, and so it will grow out, or it will be a clitoris influenced by estrogen, and it will grow in. That's the erectile tissue. So that's what that is. Okay? Now you know, and you can speak intellectually at Christmas when everybody's asking you. You're like, hey, guess what I found out? No, don't be that person. Okay, in our last situation here, I have that egg that had 2 X's fertilized by a female sperm. So this female here has what we call triple X syndrome. What's going to happen to 2 of these? They're going to be randomly inactivated. Yes. And ultimately, this female is not like some super female because she's got 3 X's. She's still a female. She still looks girlish. She may have some mental incapacities, but that's basically it. Okay? So, when we talk about aneuploidy with sex chromosomes, here's some different outcomes, potential outcomes. Now look at these conditions. Look at the title. Tell me what I'm dealing with. Analyze that title, please. What chromosomes am I dealing with? 1 through 22. Why did I not ask you what cells I'm dealing with? It's in all of your cells, and this is clearly labeled autosomal. I told you the focus of today is chromosomes. So these are autosomal, 1 through 22, but it's recessive. So what can you tell me here? I either have 2 small letters or I don't have this condition. So that means I got the recessive allele from both parents. What if I have the dominant one? I don't have this. These diseases on this list here are only associated with being autosomal recessive. If you have a capital letter or the dominant allele, you do not have this disease. So, albinism you're already familiar with. Attached earlobes is not really a disease. Cystic fibrosis we are going to talk about briefly. We've discussed sickle cell disease. Galactosemia is talking about the metabolism of galactose. PKU, this is something we test for in the hospital now, and we can usually take care of way, way early on. So this isn't usually a problem. I mean, it's a problem, but we can figure it out much quicker now. Tay-Sachs we've discussed in a previous lecture, and those offspring expire usually 2.5 to 3 years old. Cystic fibrosis is an autosomal recessive condition. I'm only explaining this because you need to be familiar for your test. But you create a mucus as a healthy individual without cystic fibrosis that has this watery consistency or viscosity. It is really important that within your respiratory cavity or your lungs that it remains a moist environment because it enables gas exchange, oxygen and carbon dioxide to pass through your air sacs or your alveoli. Individuals with cystic fibrosis do not have that runny viscosity with their mucus. Their mucus is dried. As a result, it tends to pile up, and as it does so, it makes it very difficult for gas exchange to occur, meaning it's very difficult for you to breathe. Individuals with cystic fibrosis usually complain of being short of breath and being tired, and that's a result of not having enough oxygen. Science has come a long way during my lifetime. So during 30-some years, there was a young man in my 2nd grade class who had cystic fibrosis, and every few hours he underwent percussion treatment. And for a 2nd grader, it was scarring to me. But they would beat his chest. And not like you're burping a baby, but they have to hit him hard. And they hit him on the front of his chest and on the back of him. And they had to do so every few hours to break up all the cakiness of that mucus. And then get him to kind of cough and get that stuff loose, or else he would die. He was a very fair-skinned kid, so he bruised very easily. It looked uncomfortable. And of course, in 2nd grade, I'm like, as we get to middle school, he starts wearing what I thought was a bulletproof vest. Turns out it's a percussion vest. So science and technology had increased. And this vest, he could push a button and turn it on, and it would do that percussion treatment and break that up for him. So, good. Things are progressing. He did eventually expire. But now we have treatments, oral treatments. We understand what's occurring, and we understand how to change the environment. You can take a pill, and we can supplement what's happening in your body. And with that percussion vest and a few other, we can help you increase your lifespan. So it used to be in the late teens, early 20s, and now we can get you to 30, 35, and I'm sure it's going to continue to increase. So cystic fibrosis. These are autosomal recessive. What can you tell me about these conditions? Dominant. Chromosomes, what? 1 through 22. Okay, and in order to have this condition, you have to have a dominant allele. So that means you either have 2 capitals or a capital and a lowercase, okay? And usually when you have 2 capitals, you get a full-blown form, and sometimes when you have a heterozygote combination, you get a milder version. Not always. That would be if it was incompletely dominant, but nonetheless. Conditions we have: free earlobes. I don't believe that that's a disease. Achondroplasia. Chondro, anything has to do with cartilage. In this case, you don't develop cartilage like the bone forms that you're supposed to, so you have a stunted growth. So this is usually a situation where we have dwarfs. Early balding in males, I don't feel like that's a disease. Campodactyly means that you have like rigid bent fingers. Curly hair is not a condition. Huntington's I'm going to touch on in a minute. Syndactyly means that you have webbed fingers. That's dominant. And then polydactyly, having extra fingers, it's dominant. I talked about that last week. It's like, we're all supposed to have, if dominant means that it's popular, we should all have flippers. We should all have webbed fingers. Okay? Brachydactyly means that you have short fingers. And then progeria is the one I started on today. That was the intro story. Huntington's disease is autosomal dominant. There are a lot of individuals who are impacted with this. The bad thing about this disease is that you don't know that you have it until you're in your mid to late 30s. What have you already done by this time? You reproduced. Okay? So you find out you have this super healthy family. I'm going to, as a specific story, and there are so many more stories to follow this. So you can Google stories, and they're countless. But you have this family of 4, mother, husband, 2 boys. The boys are older, high school, getting into their higher ages, not higher, but like adult ages. Dad begins to show symptoms of Huntington's disease. Very active family, very active. Dad starts tripping and stumbling, and within a year progresses to where he can no longer control his skeletal or smooth muscle. So he can't control when he pees, when he poops, he cannot walk. He is bedridden, and shortly after, he makes his way to a nursing home where his wife and sons are visiting him daily, and he expires usually within about 2 years. He expired, this is a specific case, within 2 years. But that's usually the progression of that disease. It's very aggressive. So you went from running and hiking and vacationing with your kids to 2 years of pure bad stuff. And then Dad passes away. They already had 2 big boys. Dad passes away. Guess what happens a few years later? Older boy starts showing symptoms. Followed up by the younger boy showing symptoms as well. Both these boys, neither one of them reproduced, even though they could. Why? They already knew. They already knew they had it. Their dad had it. It's dominant. They had to have gotten it. Boys slow down. They make their way to a nursing home. Mom is visiting every day, taking care of them, goes home at night, comes in one day and shoots both of the boys before they expire on their own. She's arrested for murder. She goes to trial. They rule for or against her? They ruled for her. She said, these boys told me, when I get this far, I already know what's going to happen. Don't keep, like if we're talking feeding tubes, like catheters, like these, you can't do anything. You're ultimately a vegetable, and when you can no longer think, then. So she didn't go to jail. She was told that for the rest of her life she cannot be the primary caretaker of anybody, and she was like, well, that really doesn't matter because I have no husband and no sons and no grandkids, so it's just me. But can you just imagine that the amount of duress and distress that she underwent in those few years, losing her husband and then both of her boys, and then being the one responsible for killing them, but as a mom thinking that she's doing the right thing. So Huntington's disease is an autosomal dominant disorder, and again, the bad thing about that is you don't realize you've been inflicted with it or that you have it until it's too late, right? And you've already reproduced. Okay? Look at the title of this slide, these conditions. X-linked recessive. Tell me what we're dealing with, please. Sex chromosomes. So I'm on set number 23. And in order to have this condition, you must have recessive. Either 2 small letters if you're a female, and if you're a male, just 1. And these are both conditions that we have discussed already, hemophilia and colorblindness. Testicular feminizing syndrome, what does that sound like? I mean, I hate to say like, use common sense, but it's a situation in which the testicles aren't fully developed. It says it's absent of male organs. It's not talking about external genitalia, like the penis. It's talking about the gonads, the internal organs. Those are the reproductive organs of the male. So, of course, because he is lacking testes, he's sterile, and so he would not be reproducing. That does not necessarily mean that he is missing his penis. Okay? So, the end of this chapter is, what do you do if you have a condition, or if you wonder if you have a condition, or if you're curious what your offspring could have if potentially you have a kid, or you're pregnant and want to know if your kid is healthy or not because you're a high risk or you're just a nosy person. You go through what we call genetic counseling. Genetic counseling has varying degrees of what information they can give you. Of course, if you're talking about potential outcomes, they can only provide you with probabilities. So if you come back and say, well, I thought you said my kid, that no, you can't do that. You can't do that. But if we already have a kid, we can test that and tell you what that individual has. So genetic counseling has varying degrees. We're going to discuss briefly prenatal diagnosis. But again, the whole purpose is to kind of figure out, hey, what do I want to do? Is my child going to be healthy or not? And and the major reason for genetic counseling to date right now, besides trying to diagnose a disease that a child may have, so we already have a child born, is you have two parents who are pregnant and they're high risk, and they decide to do a genetic test because they want to know early on if that child is going to have special needs, because they're not interested in raising a child with special needs. So they will choose to abort that pregnancy and not carry it full term. So those are the type of decisions that parents are making with these sciences. And a lot of people are like, oh my gosh, that's horrible. These are horrible people. But the reality is, I believe you shouldn't judge until you're in that case. And I may not like it to hear that, but I'm also not the one raising that child. And I'm not, do you see what I'm saying here? So it's really quick for society to pass judgment on people. So what we have in this situation, we have prenatal testing. Prenatal means that my mother is not my mother, but the mother that I'm testing is pregnant. Okay, that would be horrible if my mom was pregnant. So we have a pregnant female, and for whatever reason, we decide to do genetic testing on her, the offspring. You don't normally just do this. If you're a 22-year-old pregnant or a 25-year-old pregnant, you don't do this. Because with these tests, there's some risks that the child will be harmed or the pregnancy will be aborted. So you really have to go and weigh your pros and cons. Like, if I'm healthy and everything's good, the likelihood is my kid is also good. But if I'm 44 and I'm pregnant, I'm really concerned that I need to be ready if my child is not going to be. That's a different situation. So what this is showing you here is we have a pregnant mom, and this is her uterus. The uterus of a pregnant female is the topmost superior organ when she is pregnant. When she is not pregnant, the uterus is very, very small and in her pelvic cavity, so very, very low, would be covered internally by a bathing suit bottom. Okay, a very small bathing suit bottom, not like the ones that have to, it's a very small organ. But when a female becomes pregnant, that uterus gets really large and it just extends up her belly. Her organs are not displaced. People are like, well, where does your pancreas go? It stays. Like, everything stays. It's a little bit more compressed, but it all stays. Okay? So that uterus is right there on the surface. So when we do this type of testing, it's very easy to access the baby because they're right there beneath the skin and muscle of that mother. So here we're doing a sonogram at the same time to tell me the position of that child. So as I'm inserting this needle, I don't hit this child. That's my goal. Now, is there a chance I could hit the child? Yes. Okay? Because that's like telling that kid not to move at all. Like, sit in the corner. You know what that kid's about to do as soon as you turn around. Like, army crawl to the next place. Like, that's what's going to happen. This needle will go through the belly and through the uterus into the abdominal pelvic cavity where that baby that is, and it's designed to withdraw amniotic fluid. It's called an amniocentesis, but amniotic fluid. What we're trying to do is get the skin cells. Because in this fluid here, the baby is losing skin cells just like you and I do. So if we can extract those skin cells have their genetic material, right? Kind of like we did with DNA extraction. And we can analyze it and figure out what their chromosomal type looks like. If we need to be a little bit more specific, we extract the cells. We do what we call a chorionic villus testing. We extract cells from the placenta, which is a temporary organ. Either way, we're getting cells with genetic material, and we're going to put a karyotype together and determine whether this child is healthy or not, and what conditions they have. Okay? On this situation here, I want you to understand that this uterus, so it's only large, of course, because she's pregnant, because otherwise it would be small. Inside that uterus, once she becomes pregnant, a placenta forms. A placenta is a temporary organ that allows the baby to exchange nutrients with the mom. And from that placenta extends the amniotic sac, and then here is the umbilical cord, and here's the kiddo. Okay, so that's the setup there. When this kid is born, all of this comes out, and the uterus begins its shrinking process. It takes about 10 months for it to get this size. It takes 18 months to return to its initial size. This is why you should not have your kids back to back, because your uterus, as it begins to shrink, as it gets to this size and you become pregnant again, guess what size it remembers after pregnancy too? This size. 18 months. Life lesson, listen to me. Or you're going to be like emailing me later on and like, can you tell me what you meant by that? I meant when the doctor says don't have sex, it means don't have sex. Because during this time you're extremely fertile. A lot of women get knocked up one after the other, and guess what she spends the rest of her life trying to do? Lose weight. And guess what she won't be able to do? I'm not the boss of you, but I'm just telling you. So whenever this woman goes into delivery, if we're going to genetically test, we'll come into this area, and we use a very fine needle so it doesn't rupture this bag. If we need to get it from the placenta, we'll get it here. When this woman goes into delivery, this cervix right here, that is, before she delivers, the size of this pin tip, dilates 10 centimeters. That's a big deal. Takes usually a day or two, maybe a week, depending on the female. It could take up to 3 weeks. Then the water breaks, the fluid comes out, the baby should be here head first. Biggest part goes first, so nothing gets stuck. When the baby comes out, we cut the umbilical cord. We go away and take care of that baby, and then the doctor comes back to the mom, pulls the umbilical cord, and then all of this comes out. This cannot stay inside. This was temporary. And it has to be removed, or infection will be followed by death. So we get rid of it. Just so that you understand the anatomy. Proceed. Yes, sir. Oh, it's very true. You ready for this? I'm about to gross you out. In anatomy, one of the very first rules is you never compare your organs to food. I do it all the time. A placenta looks like a ribeye. A steak. It's like that size, full term, right? It's thick. Like they plopped it out on the table. Not mine. But I was at UT Southwestern, and there was a baby who expired in utero, and it's because the placenta had calcified. It had turned into bone. And, part of it had. Anyways, they put the placenta down on the table so that we could analyze it. And I was like I'm like touching it, and it's a steak. And so what some people do, because it's supposed to be so nutritious, they save it and eat it. I don't do that. But I'm not saying it's wrong if you do. But yes, sir, that is very true. Additional questions? How did it get bone? I don't know the specific answer to that case. But what happened was there was too much calcium. So I don't know if mom took in too much calcium or if she had a genetic disorder that it created but the placenta had calcified. So the baby wasn't able to get oxygen from the mom, and so baby died. So they were removing the baby, and that's how I saw it. I didn't, at that point in time, I wasn't where I'm at mentally, like where I could understand stuff. But I remember thinking, oh my gosh, because you could see the dead baby, which was super tiny, and then you just saw this big steak. And I was like, what is that? Yeah, that's her placenta. That's crazy. But that's unfortunate that the baby died, of course, and I don't know the mom. And I'm not trying to make fun of her, but it was a really cool situation to where I could learn. So, additional questions. All right, that is the end of that.